So this is our experiment on DNA and genes. And this is our introduction. So what is PV92? PV92 is the term used as PV92 alleles. Typically refers to a specific genetic marker used in population genetics and forensic DNA analysis. So we can see here in this picture that this is the location for the PV92 allele, and these are two homologous chromosomes, and each gene locus has a particular form of the gene or allele, and we will know more about the ALU element later in the slides, but this is just to make you oriented with what PV92 is. So what is ALU and how does it work? ALU elements are a type of short interspersed nuclear element, also known as sign, found in the human genome. These elements are short sequences of DNA, around 300 base pairs in the length, that are repeated hundreds of thousands of times throughout the human genome. And how they work is that ALU elements are a type of DNA sequence found in the human genome that can replicate and move around within the genome, and they work by being transcribed into the RNA, which is then reverse transcribed back into the DNA and is inserted into new locations in the genome. This process is known as retrotransposition, allows ALU elements to increase in number and spread throughout the genome. And this is how we can see how the ALU element works in the genome for hemophilia and colon cancer. So what are the different genotypes present in populations? In a population, the genotypes present at the PV92 locus would depend on the frequency of the alleles within that population. So the PV92 locus can have three possible genotypes: the plus plus, which is also known as the homozygous insertion. So individuals with this genotype have the PV92 insertion on both chromosomes at the locus. And the other one is plus minus, which is known as heterozygous. Individuals with this genotype have one chromosome with the PV92 insertion and one chromosome without it. And the last one is minus minus, which is also homozygous, but it's like the absence of the PV92 allele, so it doesn't have the PV92 allele on both the chromosomes. So our aim for the lab. What was our aim for the lab? The lab experiment aimed to determine individual PV92 genotypes. The objective was to identify whether participants possessed a homozygous dominant, that is plus plus, heterozygous plus minus, or homozygous recessive minus minus genotype at the PV92 locus. Some practical skills were acquired during the experiment, included preparing agarose gels, conducting electrophoresis, and interpreting DNA banding patterns. The experiment provided an opportunity to gain insights into the personal genetic makeup by identifying the PV92 genotype. So what was the source of DNA and how did we isolate it? So the epithelial cells contained our DNA when we swooshed the saline in our mouth, and mouth rinsing with 10 ml of saline. So what was the source of DNA and how did we isolate it? The epithelial cells contained our DNA when we swooshed saline in our mouth rinsed with 10 ml of saline solution for 30 seconds to collect epithelial cells. And then we transferred 1,500 microliters of the collected solution into a centrifuge tube, followed by centrifugation at the full speed for 90 seconds, and pipetting 30 microliters of the supernatant liquid into a PCR tube and added 100 microliters of Chelax and placed it in the PCR tube into a cycler for 10 minutes and shaking it for 5 seconds afterward. transfer of the mixture into a new microcentrifuge tube for centrifugation at high speed for another 90 seconds, and then addition of 22.5 microliters of primer and 2.5 micrometers of DNA from the microcentrifuge tube into a new PCR tube. And what is gel electrophoresis? Gel electrophoresis is a laboratory technique used to separate and analyze DNA, RNA, or protein based on their size and charge. So it involves the movement of charged molecules through a gel matrix in an electric field. So what is PCR and how is it used to target PV92 or ALU? And what's the scheme and what are the primers and why are they important? PCR or polymerase chain reaction is a molecular biology technique used to amplify a specific region of DNA. So it allows researchers to make millions of copies of a particular DNA sequence starting from just a small amount of DNA. So to target the PV92 allele or ALU insertion site using PCR, specific primers are designed to anneal to sequences flanking the PV92 locus. So these primers are short, single-stranded DNA sequences that are complementary to the DNA sequences adjacent to the PV92 site. Primers are short sequences of nucleic acid that mark the beginning of DNA synthesis. So the PCR scheme involves the steps like designing primer, DNA extraction, PCR setup and cycling, and amplification and analysis of presence or absence of PV92 insertion. And primers are crucial components of PCR because they determine the specificity and efficiency of amplification reaction. And this is a PCR machine, and this is a polymerase chain reaction.
Okay, so when DNA becomes separated by size due to the gel electrophoresis, they appear as bands in the agrogel. And there are two possible alleles that we can get from these bands. The first one is the positive one, and the second one is the negative one. The positive one means that there's an allele per transposable element, and the negative one means that there's an absence of the allele transposable element. Based on this, so we can get three types of alleles: homozygous, dominant, which is plus plus; homozygous recessive, which is minus minus; and heterozygous, plus and minus. When it's homozygous, it means that it contains two allele transposable elements, and when it's heterozygous, it contains neither. And when it's heterozygous, it contains at least one. No, it contains one. Okay, so this figure shows the photograph, and this is the photograph of the same gel containing genetic lab PCR samples. So based on this, we can determine which one is homozygous dominant, which one is homozygous recessive, and which one is heterozygous. How do we know when it's homozygous dominant? We can see that there's a band in 731 BP along line of 731 BP, and when it's homozygous recessive, there's a band that is approximately even with 328 here, 383 BP here. And when it's heterozygous, it shows two prominent band, one that is almost approximately at the same as the homozygous dominant, and this next one as homozygous recessive. So there's in this same gel, we can see one heterozygous result. This one is the first band, and here it can be seen the second band. So this is the table, the data containing our class results. As it can be seen, most of the results are unknown, but based on the results that we have gotten, we can see that there's two homozygous and two homozygous dominant, and two homozygous dominant, two homozygous recessive, and one homozygous heterozygous. This is the table, this is the graph depicted based on the above table. Lastly, both our results were gotten. The analysis that I did on myself is that the result that I got for myself is 2 positive homozygous dominant. This is, so it means that I have inherited it at least from one parent or 2 parents. It could be either way. So according to my result, I checked in the frequency of allo insertion, and based on our research, I am closer to the Nigerian, which is 0.09. But the thing is, that's a small frequency to get a homozygous dominant. So we were able to come up that it might be because I'm from East Africa, and Nigeria is West Africa, and that could cause some differences. Samiha's PCR result indicated that she was heterozygous, and we connected her to Indian Muslim here, which is 0.30. And according to the PV92, her frequency seems probably likely that it should be heterozygous. And it means that she inherited the allo insertion only from one parent. What were we able to conclude based on this? We concluded that there's 2 homozygous dominant, 2 homozygous recessive, and 1 heterozygous. Although some of the results were unknown, we can still see there's like different results that we got here, and it suggests that it has a genetic, the allo element has a genetic diversity, meaning that it is polymorphic. Our limitation in this experiment was that although the data taken was from a diverse group, it's still a small data, and it's hard to determine an entire population based on our class data. And the second problem was that 15 out of 22 data were not interpreted because no band was observed in the PCR sample, and our results could also have been affected by the insufficient primary concentration, possibly. In the future, we would like to draw a broader conclusion so that we can collaborate with other classes, and this will give us more accurate results. And the second one is we would like to explore the evolutionary implications of allo insertions, since allo insertions are based on, since they are hereditary. Do we have a thank you slide? Thank you.
Reference.
Oh, and this is our reference.